Good morning, everyone, and thank you for being here today. I'm Ananya, and today I'll be presenting my research on how knocking out Notch1 changes gene expression in melanoma cells. For background, melanoma is the most aggressive form of skin cancer because it has a very high ability to spread throughout the entire body, making this both deadly and hard to treat. Melanoma progression is driven by several major signaling pathways within the body. This includes the MAPK pathway, which is commonly activated by mutations in the BRAF or NRAS, the PL3 AKT pathway, which are immune signaling pathways, and the Notch signaling pathway, which is what I'll be talking about. One of the biggest challenges in treating melanoma is that melanoma cells are highly adaptable. This means that they can change their behavior depending on environmental conditions, stress, and treatment exposure. In particular, melanoma cells can switch between a highly proliferative state and a more invasive state. This flexibility helps tumors survive treatment and contributes to metastasis. Understanding what controls these transitions is an important area of melanoma research and connects back to a signaling pathway. A concept that has become increasingly important in melanoma biology is something called cell state plasticity. Rather than existing in one fixed state, melanoma cells can transition between different transcriptional states. On one end of this transition is the proliferative state, and in this state, cells divide rapidly, have active metabolism, and show high levels of cell cycle activity. On the other end is the stress adapted or invasive state. In this state, cells focus less on growth and more on survival. They activate pathways focused on migration, tissue remodeling, stress adaptation, and therapy resistance. This ability to switch between states allows melanoma cells to adapt to changing conditions, and this is one reason why melanoma can be so difficult to treat. So, researchers are now interested in identifying the genes and signaling pathways that regulate and control these transitions between states. My project focused on one of those pathways, specifically the Notch signaling pathway. Notch signaling is a form of direct cell to cell communication. Unlike many signaling pathways that use molecules traveling through the bloodstream, Notch signaling requires neighboring cells to physically interact with one another. When a Notch receptor on one cell binds to a ligand, such as a jagged or delta like protein on a neighboring cell, part of the receptor is released and travels into the nucleus. There, it helps regulate gene expression. Under normal conditions, Notch signaling plays an important role in cell communication, cell fate decisions, tissue organization, growth control, and development. The specific receptor I studied within the Notch signaling pathway was Notch1. In melanoma, evidence suggests that Notch1 may support tumor progression by promoting proliferation, survival, invasion, and therapy resistance. However, researchers are now increasingly believing that Notch1 can do more than simply cause growth. It can also act as a regulator of melanoma cell state plasticity. This brings us to the main question of my study. The research question I investigated was, how does Notch1 knockout alter melanoma transcriptional programs? To answer this question, I used CRISPR gene editing to disrupt Notch1 and then measure changes in gene expression. My hypothesis was that loss of Notch1 would suppress proliferation related programs, including genes involved in the cell cycle, DNA replication, and metabolism. At the same time, I predicted that stress response pathways would become activated, including immune signaling, inflammatory pathways, and extracellular remodeling programs. Overall, I expected melanoma cells to shift away from a proliferative identity and towards a more stress adapted state. To test this hypothesis, I used two human melanoma cell lines, 8375 and SKML190. My workflow began by culturing the cells and then using a CRISPR Cas9 construct designed to target the Notch1 gene. After transfection, puromycin selection was used to enrich the cells that successfully incorporated the CRISPR construct, and then RNA was extracted from the surviving cells. The gene expression of these cells was analyzed using the NanoString pan-cancer IO360 panel, which measures approximately 770 cancer-related and immune-related genes. Finally, expression levels in the knockout were compared with wild-type control cells to identify the real transcriptional changes occurring with Notch1 loss. However, it's important to say that even though both cell lines were initially included, only the A375 cells survived secondary selection and produced enough material for transcriptomic analysis. The results revealed widespread transcriptomic remodeling following the Notch1 knockout. Out of the 770 genes analyzed, 157 genes were upregulated, 127 genes were downregulated, and 484 genes showed little or no change. This means that nearly 300 genes changed expression following the disruption of Notch1. But what's more important is that the changes in genes were not random. Many of the downregulated genes belong to pathways involved in proliferation and genome maintenance, while many of the upregulated genes were associated with stress responses, immune signaling, and extracellular remodeling. This indicates a coordinated shift in melanoma cell behavior rather than a random one. First, let's take a look at the genes that decrease in expression, or the downregulated genes. Several important cell cycle genes were strongly downregulated. This includes CCNB1, CDC20, UBE2C, and MKI67. These genes play critical roles in cell division and are commonly associated with rapidly proliferating cancer cells. I also saw decreases in genes involving DNA replication and repair. This includes RRM2, MHH2, and XO1. These genes help maintain genome stability and accurate DNA replication. In addition, metabolic regulators such as BCAT1, EZH2 were also reduced. Together, these findings suggest that Notch1 helps support a proliferative melanoma state characterized by active cell division, DNA replication, and metabolic activity. So, when Notch1 is lost, these programs actively become suppressed. Next, let's take a look at the genes that increase in expression most dramatically, or the upregulated genes. One of the most notable findings here was the dramatic increase in IL-24. IL-24 is associated with stress signaling and can promote apoptosis, or programmed cell death, in cancer cells. Another major increase occurred in CDKN1A. This gene functions as a cell cycle transport regulator and is typically activated during cellular stress. Several stress response genes were also elevated, including DUSP5 and EGR1. These genes are linked to MAPK and ERK signaling and are usually activated when cells respond to stress or changes in their environment. Immune-related genes were also increased, including HLA-B, HLA-C, and CD70, which are involved in antigen presentation and immune signaling. Finally, genes involved in remodeling and migration were also strongly upregulated. This included SPP1, WNT5A, ETGA1, and ETGA2. These genes are specifically associated with extracellular matrix interactions, tissue remodeling, and invasive behavior. Together, these results suggest that Notch1 loss activates stress response, immune-related, and remodeling programs within the cell. So, when we put all of the results from the upregulated and downregulated genes together, we see a clear pattern. Notch1 appears to help maintain a proliferative melanoma state. So, when Notch1 is disrupted, genes involved in cell division, DNA replication, and metabolism decrease in expression. At the same time, genes involved in stress adaptation, immune signaling, and tissue remodeling become even more active. So, instead of shutting down, the cells reprogram themselves into a completely different transcriptional state. And if you remember, this is consistent with the concept of melanoma cell state plasticity that I discussed earlier, with melanoma cells shifting between different transcriptional programs. All in all, these findings imply that Notch1 functions as a regulator of melanoma cell identity, helping determine whether cells remain focused on growth or whether they transition into a stress-adapted state. So, to summarize all of the key findings of this presentation. Firstly, of the whole 770 genes that the nanostrain analyzed, 127 genes were downregulated and 157 genes were upregulated, directly because of Notch1 disruption and knockout. This loss of Notch1 reduced expression of genes involved in cell division, DNA replication and repair, and metabolism. At the same time, Notch1 loss increased expression of genes involved in stress responses, immune signaling, extracellular remodeling, and migration. Overall, these results reinforce and support the idea of Notch1 functioning as an important regulator of melanoma cell state plasticity and appears to help maintain a proliferative melanoma state. So, initially, rather than acting as only a growth promoting gene, which was initially thought, Notch1 appears to influence how melanoma cells specifically adapt and respond to stressful environments. These findings contribute to our new understanding of melanoma biology and may help identify new and different directions for future therapeutic research. I would also like to express my appreciation for Miss Anna Felt at Brooker Spatial Biology in St. Louis, who has given me priceless support and mentorship throughout this entire project. And also Miss Sarah Zimmerman at Sorolis Lab at WashU Medicine, who donated me the 8375 and SKML melanoma cell lines for my experimentation. Thank you for listening.